Before starting any of your processes, you need to ensure that you've undertaken hand decontamination through hand washing, and that you're wearing all the required personal protective equipment that is appropriate for the task you're about to undertake. These will be listed in your standard operating procedures and will include items like lab coats, gloves, goggles, and masks as needed. Once you've finished with the hand wash and put on your PPE, you'll need to prepare your work areas. This includes taking out any of the consumables that will need to be brought to room temperature, as well as cleaning down your workspace. You'll also need to do things like prepare disc pots that contain 10% bleach. Your workspace must be cleaned at the start and end of every day, as well as in between changes of procedure when you'll be working with different specimen types. The same process will also need to be undertaken if there are any spillages. For fluid spillages, you'll need to follow local guidelines, as there may be additional steps that you're required to undertake. Preparing and cleaning down of your workbench includes not just cleaning down the bench space itself, but any of the equipment you're likely to use, including racks and pipettes. This video covers setting up e-tests for antibiotic sensitivity testing for the pure isolates grown from either your antibiotic sensitivity plate or from secondary stroke purity plates. Make sure that the agar plates that you're using will have been brought to room temperature and completely dry. This is key to ensuring an even bacterial lawn when inoculating. You'll be using 2 different types of agar to set up sensitivity testing. For the Streptococcus pneumoniae, you'll be using Muller-Hinton agar with 5% defibrinated horse blood and 20 milligrams of beta-NAD. For E. coli, you'll be using Muller-Hinton. It's crucial to use the correct agar and incubation conditions and times when undertaking e-test testing, as over or under incubation can impact on the accuracy of the readings. You'll need to set up control plates every time you set up an e-test plate in order to help troubleshoot. And you'll need to allow the e-test strips to reach room temperature prior to opening. Make sure that the strips are protected from moisture, heat and direct exposure to strong light at all time. Ensure that moisture is not penetrated within the packaging or the storage container. Once the strips have reached room temperature, ensure that the moisture condensation on the outer surface is evaporated completely before opening the packs. Now onto the process. The process is basically identical to that you'll undertake to set up an antibiotic resistance test. You need to label a suspension tube with the specimen number and isolate ID for each isolate to be tested, as well as the one for the control strain. This will be ATCC 49619 for Streptococcus pneumoniae, or for E. coli ATCC 25922. Make sure your suspensions of test and control isolates are from fresh overnight growth on non-selective medium. This will be the AMR plate for your test isolates, as long as you are certain they are not contaminated or mixed. You will select from your control plates for E. coli and Streptococcus pneumoniae. In some cases, you will not have sufficient resistant colonies to make a bacterial suspension direct from the AMR plate, and so you'd need to make a fresh purity plate prior to this process, which is what we are going to show in this video. If you have a suspension of less than 16 minutes old from setting up an antibiotic sensitivity plate, you can mix well and use the same one. If not, make up a fresh suspension. Make your suspensions by suspending colonies in saline using either a cotton swab or sterile loop. When making your suspensions, use 2 to 3 morphologically similar colonies to avoid atypical variants, and mix the suspension by inversion. You'll now need to standardise your suspension to a density of 0.5 McFarlane standard using the Remmel-McFarlane equivalence turbid standard set. In this video we have different kits available to us, but the principle is the same. Invert the McFarlane equivalent turbidity standard gently to fully suspend the polystyrene microparticles, then compare the turbidity of the bacterial suspension. Note the bacterial suspension tube should be of a similar diameter to the McFarlane equivalence turbidity standard tube. You need to make sure you do this in adequate light And the match suspension is when equal obliteration or distortion of the black lines is present. You may need to adjust your turbidity of the suspension by either adding more organism or more saline to achieve the 0.5 standard. It's crucial that the density of the inoculum is neither too dense or too light, as this will alter the zones of inhibition and thus the antibiotic resistant reading. Once you've made the suspension, it must be used within 60 minutes or a fresh suspension will need to be made. It's optimal to use it within 15 minutes. If there's a delay, post-preparation, the suspension should be mixed by inversion prior to use. Now, once the suspension is made, you will progress to inoculating the room temperature agar plates. Plates must be labelled with a permanent marker with the bacterial isolate ID, specimen number, and AMR or E-test to indicate that this is an E-test plate. The control plates must be labelled either ATCC 49619 control strain for Streptococcus pneumoniae, or for E. coli use ATCC 25922. Labels can also be printed if this is an option. You dip a sterile cotton swab into the mixed suspension and remove excess fluid by turning the swab against the side of the corner. This is important to avoid over-inoculation of the plate. Then spread the inoculum evenly across the entire surface of the plate by swabbing in 3 directions. Take particular care to ensure there are no gaps between streaks. Once this is done, check the inoculated agar surface is completely dry before applying gradient strips. Strips can be applied to the agar surface with single-use forceps or flamed metal forceps. Position the gradient with the MIC scale facing upwards towards the opening of the plate and the concentration maximum nearest the rim of the plate. Ensure that the whole strip is in complete contact with the agar surface. Do not place the strip upside down, as no inhibition ellipse will form, since the antibiotic will not diffuse across the non-porous plastic strip. If air pockets are seen under the strip, remove them by pressing gently on the strip without moving it with the applicator tip or forceps, working from the lowest concentration upwards. Small bubbles will not affect the results. Once applied, however, the strip cannot be moved because of the instantaneous release of antibiotic into the agar. Once the strip is applied, incubate the plates in an inverted position in stacks no higher than 5 at 34 to 36 degrees C for 16 to 20 hours for E. coli and 34 to 36 degrees C in a 5% CO2 incubator for 18 to 20 hours for Streptococcus pneumoniae. You will then need to clean down your bench space appropriately.